spectroscopy. In today's session, we will continue our discussion on the basic aspects of instruments used for molecular spectroscopy. And today we will take up the detectors and the types of instruments used for the purpose. Today's session is planned as follows: that quickly we will try to recall what we did in the previous session. Thereafter, we will again recall what are the essential components of instrument used for spectroscopy studies, and in this our understanding of things will start with the detectors, followed by the data output devices. What kind of devices are used for taking out the data? How do I get a spectrum what I want to make use of? And then we will take up different types of absorption instrument used for the purpose. In this context, we will talk about 2-3 different types of instruments used, and towards the end we will sum up what we do today. Now let us try to recall what we did in the previous session. We started with recalling the basic aspects of spectroscopy. That is, spectroscopy is a study of interaction of radiation and matter, and in this process we made a case for the requirement of an instrument to make such kind of a measurement. That means, if I want to make a measurement of the interaction of radiation and matter, I must have to have a device for doing this. In this context, we outlined that there are certain essential components required for the instruments to be used for the purpose, and we realized that there are 5 essential components. One is you have to have a source of radiation. Thereafter, I must have a device called as a monochromator or a wavelength selection device, which is going to kind of take out some portion of the wavelength radiation from the polychromatic source outcome. So that was the second thing. And thirdly, we had to have some kind of a sampling device wherein we keep our matter, the thing which we want to study, to hold in that one, that is sample holder. And thereafter, we have to have the detector which will give us, which will try to tell us about whether the radiation is absorbed or not, if yes, to what extent, if it is scattered or emitted. So all those things are picked up by the detector. And towards the end, we need some kind of an interface which can give the output of the detector, which basically is going to convert our the photon, the radiation which is falling on the some other device there, into some kind of electrical signal. That electrical signal has to be converted into a suitable output which we can make use of. So that is the kind of essential components we realized. And thereafter, we talked about the source of radiation to be used for spectroscopy measurements in different regions. That means, what do I need in case of a microwave region, or in IR region, or in UV visible region. So we talked about all kinds of sources which are used for the for creating instruments to make measurements in different regions of the EM spectrum. Then we explained the principles of the type of devices used for wavelength selection. Therein we talked primarily about the monochromators, and we did talk something about the filters as well, which are the crude devices used in visible spectrometers. And in the context of monochromators, we talked about 2 types of monochromators, that is the prism based monochromator and the grating based monochromator. And thereafter, we discussed the sampling handling devices used for different purposes, and we realized that the basic premise of all this is that the material we use for sample handling device must be made of such a material which is transparent to the kind of radiation being used. So having done all that, we move on to the detectors. That means, from a source of radiation, a polychromatic radiation is coming, I put a monochromator, check out the kind of radiation I want to make use of, make it fall on the sample, and then thereafter what has happened as a consequence of that I want to make measurement of. So I need some kind of a detector for the purpose. So we can define a detector basically is a device that converts a light signal to an electrical signal, and that is suitably measured and transformed into some kind of an output. So that is the role of a detector. And typically there are 3 types of detectors we used, and these happen to be a phototube, photomultiplier tube, and diode array detector. So there are 3 types. We learn about each one of them in small bit of details over here. Now we start with phototube. Phototube, as the name suggests, is a device based on photoelectric effect, wherein what we do is we take an evacuated glass chamber here, wherein we have got an anode here and there is a cathode. So these two are put into a evacuated quartz window, and then there is some kind of a since it is a quartz window, the radiation which is coming, which we want to detect, that comes and falls on this cathode and emits electron from there. And these electrons, because of the potential difference between the two electrodes, they are moved towards the anode and generate what is called as a current. Okay. So, that is a photoelectric current. So, the magnitude of this current is proportional to the intensity of radiation which is falling on that, and that is suitably converted into an output there. That means, the current which is coming as a consequence of that is measured, amplified, and then suitably converted into an output. So, that is a very simple device. The next one is photomultiplier tube. As the name suggests, it is a photomultiplier. That means, it is going to do some kind of an amplification of the input here. Let us see how does it work. Basically, a photomultiplier tube consists of a series of electrodes called as diodes. So, what you see in this diagram here, this is a schematic diagram. So, these curved things here are the diodes, and in this photomultiplier tube, these diodes are kept at successively higher potential. That means, diode 1 will be certain potential, number 2 will be at higher potential, and so on and so forth. That means, as I move say from here 1, 2, so and so, there will be number of them there. I am just showing 4 or 5 of them. So, each successive diode will have a higher potential, and the reason for that is that when an electron enters, say, because I am trying to measure a radiation, when the radiation comes, the photon is coming, not electron. When the photon comes, strikes this cathode here, and it emits an electron from here. When it emits an electron, because of the potential difference between this cathode and this diode, this electron moves towards the diode. From here, this will generate the secondary electrons, and they will be further reflected, put onto the next diode, and so on and so forth. Since the successive diodes are at higher potential, so at every stage one electron goes, there are number of secondary electrons are generated there. So, when these fall on the next one, still more number. So, this number goes on increasing. So, when the output comes here, so that happens to be much larger in intensity. So, essentially the role of a photomultiplier tube is it multiplies the output, and as a consequence the final output happens to be much larger than the initial signal there. So, even if the signal happens to be weak, photomultiplier tube gives you a reasonably measurable signal there, so that we can get a good spectrum from there. The third type of detector we use are called as diode array detectors. These are semiconductor based detectors. So, what happens is, in case of such detectors, as the name suggests, diode array, so that means a arrangement. Okay. So, in this case what happens is, you take a number of silicon diodes and arrange them on a chip. You take a very fine small chip, on that chip a number of diodes are arranged sequentially like this. As you know that these diodes are such, when some radiation falls on that, they are basically photodiode in that sense of it. So, they will become more conducting, and the increase in conductivity of a given diode is proportional to the intensity of radiation which is falling on that. Now, the basic premise here is that in this case of a detector, you have a chip on which there are large number of diodes are put onto that chip there. Since there are large number of diodes over there, so what can happen is, so when the radiation is falling, simultaneously that different kind of radiations can be measured in one go. In other cases, what we talked about, the phototube and photomultiplier tube, there is one particular kind of a photon coming there, striking there, electron generation is there, generates a current, that is amplified. So, that is one by one we are trying to detect them. In case of a diode array, what happens is, we can simultaneously detect a large number of different wavelengths spread over a range. So, that actually makes instrumentation, make measurement faster and easy to handle thereafter. In today's scenario, such detectors are actually are detectors of choice, because that really save the time of our measurement over there, because we can even the instruments are getting modified in the light of these kind of detectors being available there. Okay. Now, having talked about detectors, so there are three types of detectors we talked about. Let us move on to what are different types of spectrometers we use. I will focus attention primarily onto the type of spectrometer used in UV visible range, because they are the most commonly used instrument, as I mentioned earlier too. In case of spectrometers, basically there are three broad types. Let us see what these types are. The first one happens to be what is called as single beam spectrometer. We will talk about that as we move. and then there is a double beam spectrometer, because this nomenclature has a significance, because they talk about the nature of spectrometer I am going to use of. And the third one is photodiode array spectrometer. Let us see each one of them one by one. I am going to give some kind of an outline, a schematic representation of how are different components arranged into to generate or to create a single beam spectrometer. So, what you see is all the essential components we have been talking about all these times are there. So, there is a source which is depicted by some kind of a bulb. Bulb is just a representation here, because that source will depend on what kind of range I am trying to look at. So, there is a source there. From the source I am getting radiation of different wavelengths. It is a polychromatic source. So, I must use some kind of a wavelength detector device, which basically happens to be, maybe it is a filter, or maybe it is a monochromator, or maybe it is a interferometer, or whatever. So, there has to be some device of this kind there. And then what happens is, after this wavelength selection, the radiation which comes out of it, it falls on essentially, let us try to put things in perspective. Typically, if I want to make a measurement, I want to take a spectrum of a given sample. What do? I take the sample. In some cases, we take the sample as such, we take the neat sample, but many times we do not take the sample as such, we make some kind of a solution there. That means, we dissolve it into some solvent, or maybe you use some kind of a buffer system for that. So, essentially, when I am trying to take a solution, putting into a radiation's path, then this radiation is going to interact with my sample, which I want to really, my analyte, which I want to talk about or want to study about, and also the solvent system there, or other things which are in the system, they all are interacting with the radiation. So, the outcome will be sum total of all these interactions, but what I need is, I need to know about the molecule I want to talk about. Say if I take say I take an amino acid tyrosine, put into water, or I take a small peptide, okay, a peptide which contains an aromatic amino acid, and I want to study that peptide. So, what do? I do not take a peptide as such, I make it to a solution and then take peptide, put into that wavelength in the path of the radiation, and then see what happens there. So, what is going to happen is, when I take that, put into a solution, when I make a solution, I use a solvent for that, typically it is water, or maybe there will be some other components in that, because typically if I want to mimic some kind of biological system, I may use some kind of a citrate buffer or things like that. So, when this interacts, so there will be lot of components which are there in the sample chamber, which are going to interact with the radiation. So, I want to kind of subtract all that superfluous thing, or the things which are not required. So, for that what we do is, there are two kind of chambers. In one of the things we put our sample, in the second one we put the reference. That means, I take two containers which are matched with each other, that means they are identical in all respects of it, that means they have got similar kind of transparency to the radiation being talked about. In one of them I take my sample, which essentially is solution of a given material to be studied, and the second is my solvent system that contains everything but the sample. So, then what do? I make this radiation fall onto, say, the reference to begin with. I take the reference, and suppose I want to study say the spectrum from say about 200 nanometers to about 350 nanometers. So, what do? I will tune my monochromator in such a fashion that I start with, I make the output only say 200 nanometer. Nowadays it is possible to just tune it, you just feed in, the monochromator will be kind of controlled with a microprocessor, we can generate that. So, what will happen? The radiation of 200 nanometers will come, fall onto the sample and see what happens over there. Then 201, 2, 3, 4, I can do that, I can tailor that, I can kind of instruct my machine to do that. So, essentially to make a spectrum, radiation right from 200 to 270 or 350, whichever I choose, that will be made to pass through the reference material one by one, and whatever the outcome comes there, whatever is coming out of there, that will be picked up by the detector over there, and that will be suitably amplified and put into some kind of a storage device, because in the computer it will be in the memory there. Thereafter, what do is, with the help of some kind of a shutter here, I remove the sample from remove my reference and bring the sample into the path, and thereafter I repeat the same exercise all over again. So, what happens now? I will be making the monochromator give out radiation starting from 200 nanometers to 350 nanometers and make it go through a sample and again generate the same amount of data. And then for each wavelength, I will subtract the reference from the sample, so that in the process what do I get? I get what is the interaction of my sample with the range of wavelengths I am talking about there. So, essentially the same beam is made to fall onto the sample as well as the reference, and the spectrum is obtained by subtraction. Let us move on to double beam spectrometer, and the name suggests we are using two beams. Actually, it is not two beams, a single beam has been split into two. Let us see what happens in this case. The first two things remain the same as before as in case of single beam spectrometer. So, we have a source, there is a monochromator there, some kind of wavelength selection device. Thereafter, when the radiations come out of that, what we do is we use a suitable beam splitting device. That means this beam is split into two parts. That means basically I am converting one beam into two beams there, and then each one of them is made to fall on a mirror. Basically, it is a matter of optics over there. So, what is done is this radiation is baked into two radiations of equal type, and then they are made to fall simultaneously onto the reference as well as on the sample. And then there are two detecting devices there, again identical to each other. So, what happens is we are saving on time, because what we did in case of single beam was that we first run the spectrum for the reference, then for the sample, or vice versa, and subtract either manually or with the help of a computer. Now, in this case, in double beam spectrometer, the monochromatic radiation is made to split and pass simultaneously through the reference as well as sample. And then, so the outcome from here, the output of the reference side and the sample side, they are processed by the computer, they are amplified and given as an output. So, in this process what happens is the difference in signal is measured in one go. So, you do not have to take two spectra and make a subtraction there, this is done simultaneously. So, this is a time saving device, and most of the instruments nowadays which come across they are double beam spectrometers, because they are very convenient to use and is more reliable and time saving devices. Now, the third one which is even more kind of in demand nowadays, that is the photoarray spectrometers. In this case what happens is the setup changes in the sense that you have a source, thereafter through a lens we make this polychromatic radiation, because it is a polychromatic source, all the radiations are made to pass through the lens and they are made to fall on the sample. That means I am not using any monochromator there, I am not trying to kind of separate radiations and make it fall on the sample, I am making all of them fall simultaneously on the sample. So, all the radiations from the source are falling on the sample through a lens, and that means each one of them will be interacting differently with the sample, and outcome of that will, that output from the sample will be made to fall on a grating there. So, you see that here that the kind of the sequencing is very different. You do not have detector right away. So, what you have is you have a grating device here, that means the monochromator is coming somewhere here, not after the source, but it is coming after the sample. Then thereafter, once these radiations fall on the grating, so they are dispersed and then they are reflected and made to fall onto the photodiode array, which you remember is a chip which contains large number of diodes on that. So, in this process each of the diodes is going to measure one different wavelength there. So, it means all the wavelengths are simultaneously measured. So, this is even more time saving a device. So, they are in demand nowadays, and that means you are making the polychromatic radiation fall straight onto sample, and then the transmitted radiation suitably dispersed by the reflection grating is then made to fall on the detector and detected simultaneously. So, that is the basic principle of our photodiode array spectrometer. Let us take up because all these three things we talked about, they are typically used for UV visible spectrometry, which is, as I have been mentioning again and again, they are most commonly used spectrometers. But let me just share with you the instrumental setup, the setup for a Raman spectrometer, dispersion Raman spectrometer. In this case what happens is, you remember that when you talked about sources, in case of Raman spectrometer typically we are using laser sources, because they are highly coherent monochromatic sources there. So, the radiation which is coming out of a laser, which is of a particular wavelength, is reflected by a mirror, is made to fall on the sample, and it interacts with the sample and gets scattered there. You remember, because the Raman spectrometry is very different from the absorption or emission spectrometry, because it's based on the scattering of radiation. So, and typically it's measured at a different at an angle about 90 degrees from the sample, because the detection is somewhere here. We'll talk more about it when we talk about Raman spectrometry particularly. We'll discuss in detail about all this, but right now in terms of the instrumental setup, what I want to bring home is that the setup is slightly different here. So, the scattered radiation is made to fall on a curved mirror, and from there it goes on to a monochromator, and then after suitable processing it goes to a detector, and then it goes for an output device. Okay. So, that is the kind of setup we have here. So, we see that the setup is different. Similarly, if you go for fluorescence measurements, things will be different there, or maybe I go for IR measurement, things are slightly different, but all said and done, our essential components remain the same. That means you have to have a source, you have to have a sample holding device, you have to have some kind of a monochromator, whether put after the source or maybe after the sample has been interacted with. Then of course, you need to have some kind of a device to detect the consequence of interaction between the radiation and matter. And of course, towards the end we will need some kind of a means, some kind of an interface which can give you an output. Okay. So, I am sure that I have been able to put before you different kind of setups we can make use of in trying to study the interaction of radiation and matter, that means the spectroscopy or the spectrometry. Now, as regards data output devices, actually data output devices have been evolving with time. To begin with, probably our eye happens to be one of the most commonly used detector there, because that is what we are gifted with. But in terms of instrumentation, if you go back in time, initially when the spectrometry came into being, we were using photographic films or glass plates to take the output over there. Then thereafter we started talking about CRTs, cathode ray tubes. So, you had oscilloscopes, so you could see the signal on that. So, that is how it came up there. With advent of technology, we came down to recording things. We had an output. Earlier we used to see something and then make notes of that and then try to make a spectrum out of that. And then thereafter, nowadays we are using microprocessors. That means you have some instrument, most of the instruments which are coming nowadays are interfaced with some kind of a microprocessor which will control what kind of ranges to be given. There are a lot of things we can play around with and do with the help of a microprocessor controlled instruments there. Nowadays we have gone even further. We have got data handling systems wherein which are very kind of comprehensive kind of systems, wherein you can take the output from the detector and then process it in any which way, and then you can do variety of things with that. So, that is how things are changing, and this is thanks to our computers getting more and more friendly and faster. So, we have got that kind of systems in place now. Let us sum up what we have done today. What we have done today is that we reviewed what we did in the previous session, wherein we recalled that what are the essential components of instruments used for spectroscopic studies. That means there are 5 things we talked about a number of times, and then thereafter we explained the principle of detectors used in spectroscopic instruments, because detectors are very crucial, because sensitivity of detector is very crucial for the outcome of that, the kind of spectrum I get, the kind of interpretations I can look into. Thereafter we described different types of absorption instruments, wherein we talked about single beam spectrometers, double beam spectrometer and photodiode array based spectrometer. And towards the end we talked something about the setup which is there, setup of the essential components for a Raman spectroscopic measurement. With that, probably that is all what we wanted to talk about as regards the instruments used for spectroscopic measurements. In the next session we will take up we will now come down to spectroscopies per se, one by one, because we talked so far we have been talking about general aspects of spectroscopy and instrumentation. Now we will move on to spectroscopies, particular spectroscopies. We will start with rotational spectrum, or so called micro